The first experiment conducted is about the reflex action. The aim of this experiment is to study the response to the knee-jerk reflex. Knee-jerk reflex is a deep tendon reflex seen as a sudden kicking movement of the lower leg in response to a sharp tap on the patella tendon. This experiment begins with subjects sitting on a stool with leg hanging, and gently tap the knee at the correct spot using the reflex mallet. The right spot to exert the knee-jerk reflex is at the patella tendon, which is located right below the kneecap, called the patella. As for the result, the leg of the subject were lifted up as a sign of response when the patella tendon is tapped with reflex mallet. This diagram illustrates a monosynetic stretch reflex and reciprocal inhibition of the antagonist muscle. Knee-jerk reflex is a form of stretch reflex which occurs via a monosynetic reflex. When the patella tendon is hit by reflex mallet, it stimulated the muscle sphincter, which is located in the quadriceps muscle. The stretch receptor is therefore activated. Sensory neuron detect the stretch signal and carries the impulse to the spinal cord, where it will synapse directly with a motor neuron. The motor neuron then sends activating impulse to the quadriceps, causing the muscle to contract, which extends the knee. This causes a contraction of the quadriceps muscle. At the same time, in the spinal cord, the interneuron transmit impulses that inhibit the antagonist muscle, which is known as the hamstring. Simultaneous inhibition of contraction of the bicep muscle occur to prevent conflict between opposing muscle, which helps to maintain posture and body coordination. This is known as the reciprocal innervation, whereby one muscle, which is the quadriceps muscle, contracts and its antagonist bicep muscle relax at the same time. An abnormality of the reaction suggests that there may be damage to the central nervous system.
The next part of the experiment is about pupillary reflex. The aim of the experiment is to find out how the diameter of the pupil changes in response to light. The experiment was carried out in a fairly dim room, and the diameter of the subject's pupil were taken before, during, and after the light was shown onto the eyes. The result of the experiment is as follows. Before the light was shown onto the eyes, the diameter of the pupil is found to be 0.5 cm. During 0.3 cm, and after 0.4 centimeters. How does this occur? The answer to the question can be explained via the mechanism of pupillary light reflex. The diagram shown here is the view of our eyes from the top. When light is shown on our eyes, the photoreceptor cell will send signal to the retinal ganglion cell, which will then convey the information down the optic nerve to the pretectal nucleus. The signal then travels down to the Edinger-Westphal nucleus here. From the Edinger-Westphal nucleus, the information is carried down via the oculomotor nerve to the ciliary ganglion. Nerves from the ciliary ganglion innervates the iris sphincter muscle, which will cause pupil to constrict. The mechanism of pupillary light reflex is a 4-neuron mechanism because it involves 1, 2, 3, and 4 neurons. The reflex is governed by the autonomic nervous system, in which the parasympathetic division causes the constriction of pupil, whereas the sympathetic division will cause the dilation of pupil.
This part of experiment is about Purkinje-Sunsen arc. The aim of this experiment is to observe the changes in the anterior surface arc of the eye lens using the method of Purkinje-Sunsen arc. The test is carried out in a dark room. A subject was seated on a chair. A candle was then lit and placed in front of the subject's eyes. The subject's eyes were then looked into. Purkinje-Sanson images are reflections of objects from the eye surface. 3 images were detected in this experiment. The first Purkinje image is produced by reflection from the anterior surface of cornea. The second image is the reflection from the anterior surface of lens, whereas the third image is reflected from the posterior surface of lens. The first image is small, sharp, and erected. The second one is larger, erected, and less sharp, while the third image, which is on the right side, is the smallest and brightest, less sharp, but it is inverted. The next part of the experiment is about color blindness test. This test is carried out to differentiate the colors. The normal test conducted for color blindness is by using the Ishihara charts. The charts consisted of a series of pictures that are made up of colored dots with scattered patterns. In the patterns, numbers and figures are made from the dots with one color, and the background is made from the dots of different colors. Our subject is required to identify the figures and numbers seen. Color blindness is the inability to differentiate the differences between certain colors. This condition is due to the absence of color-sensitive pigment in the cone cell of the retina, which is the nerve layer at the back of the eye. Humans have 3 kinds of color receptor cells, and each of them contain a different visual pigment. For our experiment, our subject was able to differentiate and identify the numbers and figures displayed in the Ishihara chart. The result showed that our object does not have color blindness problem.
Part C. Vision Acuity Test. The aim of the experiment is to evaluate the acuity of vision of the subject. How do we do it? Step 1. Subject must be 6 meters away from the chart, and each test is carried out with one eye closed. Step 2. Examiner will point at the chart and the subject is required to read out the alphabet. Step 3. If the subject made few mistakes, stop the test and jot down the last row that can be read properly. Results. The vision acuity of our subject is 6/18 for right eyes and 6/6 for left eye. The normal visual acuity is 6/6. Discussion. Visual acuity is the acuteness or clearness of vision. It is also a quantitative measure of the ability to identify black symbols on the white background. Since the subject has visual acuity of 6/18 for right eye, the subject is having myopia, and concave lenses is needed. Part D. Ophthalmoscopic examination. The aim of this experiment is to examine several structures of the eyes. The tool that we need in ophthalmoscopic examination is an ophthalmoscope. How do we do it? Step 1. Ophthalmoscope is positioned onto the examiner's eye and examination is started at approximately 1.5 feet from the subject with a suitable light intensity and focus on the pupil. Reflex of red light originated from sclera, choroid, retina, optic disc, and blood vessel is observed. Step 3. The ophthalmoscope light is directed to the nasal part where the optic disc will appear whitish in color. Step 4. Macula is located toward the temporal optic disc. The blood vessels in the eyes do not contain any pigment. What are actually seen will be the blood column. Result. The red light reflect is observed before approaching nearer to subject. Blood vessel and macula could not be identified. However, optic disc can be seen. Discussion. Ophthalmoscopy allows eye fundus, which is the back part of the eyes, and other eye structures to be observed using an ophthalmoscope. Ophthalmoscopy is done as part of routine physical or complete eye examination. In the experiment, macula and blood vessel cannot be detected because of lacking experience when handling ophthalmoscope. What is diopter? It is a unit of reflective power equivalent to the reciprocal of the focal length of a given lens. What is accommodation? It is the process by which eye change focus. Ciliary body around the lens changes shape of lens, allowing eyes to focus at various distance.
This part of the experiment is about audiometry test. Audiometry is a hearing test which is carried out using pure tone audiometer. The aim of this test is to identify and diagnose the degree of hearing loss at any stage. Pure tone audiometer consists of equipment that produces sound wave and is connected to the headphone through a sound intensity controller. This test must be done in a quiet room as noises will increase the threshold value of hearing. Blue color of the headphone will be for the left ear and red color will be for the right ear. We also have here what we call as patient response button. Subject will be asked to hear the sound wave produced by the pure tone audiometer through headphone. Every sound that is heard by the subject, button will be pressed to indicate they heard the signal, and the light on the audiometry will be lightened to let the examiner know they heard the sound and be recorded in audiogram paper. Let's look at the audiogram. Audiogram is a graph used to document the degree of hearing loss. We record loudness on the left side of the y-axis with unit of decibel from 0 to 120 decibel, with 0 being very soft and 120 being very loud. And on the x-axis is the frequency from low to high frequency from left to right. We start the test with frequency of 1000 Hz and the hearing loss control knob is turned to 50 dB. We can know the degree of hearing loss by looking at the graph. Normal hearing is anything at or above 25 decibel. A mild hearing loss is range from 25 to 40 dB. Moderate hearing loss is from 40 to 55 dB. Moderately severe hearing loss is from 55 to 70 dB, while severe hearing loss is from 70 to 90 dB and profound hearing loss is recorded at anything at or below 90 dB. The result obtained is as follows. X mark indicates sound heard on the left ear of the subject, and circle mark is sound heard from the right ear. Based on the result obtained, it is observed that in overall, the subject right ear and the left ear is almost the same. Both ears able to detect the sound with the same frequency from the audiometer at lower tone intensity. Our subject can detect tones in a range of 500 Hz to 6000 Hz at a hearing level of 15 dB to 35 dB. Our subject falls into the category of normal hearing at 2000 Hz to 6000 Hz and mild hearing loss at 500 Hz to 1000 Hz. As for the conclusion, subject is known to have normal hearing.
This part of the experiment is about the otoscopic examination. The aim of the experiment is to visually examine the external ear, which includes the external auditory canal and the tympanic membrane, using an otoscope. The ear of the subject was pulled up and out to straighten the S shape of the ear. This will ease the examination of the tympanic membrane. The biggest otoscope speculum was chosen that can fit into the external ear canal. Otoscopic examination was proceeded once the speculum has been inserted properly into the ear as this process requires full concentration from the examiner. In the experiment, we were able to observe the external auditory canal, cerumen, tiny hairs, and also the tympanic membrane or eardrum, which appears as a grayish shiny screen. The otoscope is a device used to examine the auditory canal for any infection or blockage. It can also be used to examine the tympanic membrane for presence of rupture or damage. The tympanic membrane is a thin, semi-transparent membrane that separates the external ear from the middle ear. In response to sound, the tympanic membrane will vibrate, and the vibration will be conveyed to the middle ear. During the observation, we also observe cerumen and tiny hairs. A combination of cerumen and tiny hairs actually helps to prevent dust and foreign particles from entering the ear. Cerumen is also known as earwax, and it is secreted by the ceruminous gland.